So I looked it up again, and Snuppy's name is from the Seoul National University puppy. So it was going to drive me crazy until I figured it out, but that's where Snuppy's name came from. So that brings us back to George W. Bush. And in 2006, the Senate actually voted to expand federal funding of embryonic stem cell research. A bill that had previously passed the House then went to the Senate, passed the Senate, went to the president's desk, President Bush, and previously he had promised that he was going to veto this, and he did. It was actually the first veto of his administration. And he believed, and what he stated in vetoing that bill was that it was crossing a moral line and would support the taking of innocent human life. And when he signed this veto, he was actually surrounded by what at that time was called snowflake children. And snowflake children then were children who were born from an embryo adoption program. So the parents had adopted these embryos, had them, they were born, and so he was surrounded by these snowflake children and their parents. And he said, these boys and girls are not spare parts. So that's where the Bush administration stood on the embryo funding. Now, in 2009, federal regulators in the Obama administration set new rules for research with embryonic stem cells. And President Obama created a panel of scientists and ethicists to ensure that couples truly consented to the use of their embryos to create stem cells. So he made it so that there was no question as to the intent of the parents with regard to these embryonic stem cells. And then the scientists and the AMA and everyone seemed to be very happy with that. Another important historical event that was important for stem cell research was in 2007 when a researcher from Japan discovered how to use four specific genes to tell skin cells to revert back to the pluripotent cells. And these were called induced pluripotent stem cells. It took a little while for the world to recognize the significance of this achievement. He and another researcher won the Nobel Prize in 2012. So, what he was able to accomplish was to take these four cells and to tell a differentiated somatic cell how to revert back to that primordial undifferentiated stem cell. So this accomplishment eliminated the need for these embryonic stem cells derived from actual embryo. So a huge discovery at that point. Another important historical event was in 2009 when researchers were able to swap the mitochondria in the embryos of monkey mothers and replace bad DNA with good DNA. So mitochondria can carry some heritable genetic diseases, such as those that cause muscular degeneration. So when the nucleus of that cell is transferred from an existing person or an ancestor, it has to be transferred into something, and that something is a healthy egg from which the original nucleus has been removed. And it turns out that these host eggs have some DNA. So this whole process, this mitochondrial replacement, represented a different kind of gene therapy than what was already existing. So these, they called this the germline gene therapy. And although some of the ethicists and scientists think that this was an ethical line to cross, champions of this therapy say that, the whole point of making the change and altering the mitochondria is to get rid of these heritable diseases, and that's beneficial for the future of all children of the descendants. So that was an important event. Another event in 2003, we had previously the birth of Dolly. Now we have the death of Dolly. She died 6.5 years after her birth, which was somewhat shorter than the typical lifespan of a lamb. So there were a lot of speculation as to what caused that. Her DNA came from a lamb that had already lived many years. So at birth, the telomeres were already short. So telomeres are that non-gene segments of the DNA at the tips of the chromosomes, and as they shorten, our cells age, and we get age-related problems. So the man who created Dolly cited a couple of reasons in speculating about why she died. First, she was overweight, and he attributed that to all of the tourists, the scientists, the reporters, all who fed her treats while they were visiting her. Second was that for her protection, she lived indoors. All the time spent standing on her hind legs, begging for treats, along with her extra weight, caused her hips to dislocate, which contributed to her death from a progressive, irreversible disease caused by a respiratory infection that sheep living indoors commonly contract. Now, she was euthanized, but she was at the point where she was not in good shape. Post-mortem, they looked at Dolly. Her telomeres were, in fact, consistent with a lamb that was much older than her 6.5 years. There was a reduction in her telomeres due to the advanced age of her donor cells that they think contributed to her death. So that was goodbye, Dolly. So there were some setbacks in the research and the viability of stem cells. So a lot of the predictions didn't come true. Those iPS cells derived from, so for example, the skin cells, they seem to retain a memory traces of being whatever they were before, so that they're not easily turned into other cells. So like using those stem cells, those cells for that had previously been skin cells for cardiac cells didn't work quite the way they had hoped. So that was one of the issues. Then there were a lot of people who were claiming that they were going to other countries to get these stem cell treatments and had successes, but they were never really substantiated claims. So those were some of the challenges. So things weren't quite panning out the way that people had hoped with regard to the stem cells.